He did not set out to save millions. He was not looking for a miracle. The story does not begin with a grand plan or a bold declaration. It begins with a simple mistake, a moment of untidiness. It starts with a dish left unwashed on a bench. Most people would have thrown it away. They would have cleaned the mess. They would have started over. But this man paused. He saw something that was not supposed to be there. In that small act of looking, history took a quiet, unexpected turn. His name was Alexander Fleming. He was a doctor. He was a scientist. He worked at St. Mary's Hospital in London. The year was 1928. His laboratory was not large or grand. Filled with the tools of his trade, glass tubes, bottles of chemicals, a microscope that let him enter the world of the very small. Fleming was a bacteriologist. He was known for being brilliant. He was a bit disorganized. He had a sharp eye for details that others might miss. He had served as a doctor in World War I. He had seen countless soldiers die from infected wounds. He knew the terrible power of bacteria and the desperate need for a way to fight them. He walked in to find the stacks of Petri dishes he had left behind. Most of the dish was a cloudy film of bacteria, as expected, but in one area a patch of bluish-green mould. All around that mould, a clear, clean circle, a ring of nothing. The bacteria had not grown there. It looked as if they had been dissolved, wiped away. Another man might have seen a ruined sample. Fleming saw a clue. He stood there for a long time, just looking at the dish. Bacteria were supposed to grow everywhere. Mold was just a nuisance, a sign of contamination. But here, the nuisance was doing something impossible. It was keeping the bacteria at bay. He did not throw the dish away. He put it aside carefully. He had a thought, a simple but powerful idea. What if the mold was making something, releasing a substance into the jelly, a juice that was poisonous to the bacteria? He took a tiny sample of the mold. He placed it under his microscope and looked. He saw the tangled threads of a common fungus. He grew the mold in a liquid broth, letting it thrive for days. He then filtered the mold out, leaving only the liquid it had grown in, the mold juice. He took this juice and added small drops of it to fresh plates. He watched, he waited. The results were stunning. The mold juice stopped the bacteria in their tracks. It killed the germs that caused pneumonia, scarlet fever, diphtheria, yet it did not seem to harm the cells of animals. He tested it on a laboratory mouse. He tested it on a human colleague. It was safe. He gave the substance a name. He called it penicillin. Fleming wrote a scientific paper about his findings. He published it in 1929. He described the mold. He explained how he had made the juice. He showed which bacteria it killed. He suggested that it might one day be used to treat human infections. He had laid the first stone on a new path for medicine. But the world did not immediately notice. The mold juice was very unstable. The penicillin within it broke down quickly. It was hard to get more than a tiny amount. Fleming was a bacteriologist, not a chemist. He tried to purify the substance, but he failed. The methods were too difficult. For years, penicillin remained a laboratory curiosity, interesting but impractical. Fleming did not give up. He kept the original strain alive, used crude penicillin to keep cultures clean, sent samples to anyone who asked. For more than 10 years, the discovery lay dormant. The world went on as before. People still died from infections. Doctors still stood by, helpless. The potential was there, locked inside a fragile liquid no one could purify. History had paused again. The story then moves to another city, to another lab. At Oxford University, Howard Florey, Ernst Chain, Fleming's old paper. They saw the potential others had missed, had chemistry skills Fleming lacked, and decided to take on purifying penicillin. They had to find better ways to grow the mold, using all sorts of containers, even hospital bedpans. They tried different chemical processes to extract pure penicillin powder. At first, only an amount as small as a pinhead, but they kept refining methods. Long hours in a poorly funded lab, racing against time. Then war came to Europe. World War II began in 1939. The need became desperately urgent. Soldiers were dying from bullets, from bombs. and from the dirty wounds they caused. The British government and later the American government realized the importance of what was happening at Oxford. They poured money and resources in. The small lab experiment became a massive industrial effort. Scientists flew to the United States with mold spores hidden in their coat pockets to save them from an air raid. Huge factories were built. Researchers searched for new, more productive penicillium strains. They found one on a cantaloupe in Illinois. This mold produced hundreds of times more penicillin. Production soared. By 1944, there was enough penicillin to treat all the Allied soldiers wounded on D-Day. The quiet discovery from a messy lab had become a weapon of war, a tool to save lives on an unimaginable scale. Soft. The world made new. The world that Fleming had known, the constant fear of infection, began to fade away. It was a revolution as quiet and profound as the discovery itself. Doctors now had a weapon. They could treat pneumonia, syphilis, gonorrhea, scarlet fever, and countless other bacterial diseases. The change was swift and dramatic. The charts in hospitals showed a steep and sudden drop. Simple things changed forever. Childbirth became much safer for mothers. Surgery, once a risky gamble with infection, became a routine procedure. Doctors could now perform complex operations, knowing they had a way to protect the patient afterward. The lives of millions were saved. The average human lifespan grew longer. The world's population began to climb. Fleming, the quiet man from the small lab, became famous around the world. He remained modest. He often pointed out that he did not invent penicillin. Nature did that. I only discovered it by accident. The chain of events he had started with a single glance at a petri dish had wrapped around the globe. Its legacy is in every hospital, in every doctor's office, in the medicine cabinet of almost every home. The discovery of penicillin spurred the search for other antibiotics, building an arsenal against bacterial disease. It changed how we treat sickness and how we think about our bodies and the microscopic world around us. A new kind of hope was born from that messy plate, that science can find answers in the most unexpected places. The story is simple. A man went on holiday. He came back to a messy lab. He noticed something odd. He asked a question. He did a test. That is all. Many people might have seen that mold. A cleaner would have washed it away. A less curious scientist would have thrown it out. But Fleming's mind was prepared by years of study and by the memory of dying soldiers. He was ready to see the meaning in the accident. These small human moments are where the great changes begin, not in a flash of lightning, but in a quiet moment of attention. This story survived because of a chain of events: Fleming's observation, the patient work of Florian Chain, the desperate need of war, the power of industry. The discovery was fragile. At any point the chain could have been broken. We built upon that discovery. We found new drugs. We fought new diseases. We have forgotten the fear.